good question come up during that session as to what this K actually means. So K is quite literally the average expression level required for a gene to be detected in half of the cells. So go across there to the curve and then down to the bottom. That will give you what K is on average. And it's actually quite useful for comparing how good your detection level has been in your data set. So if you have a very high value of K, you've got quite poor detection. Whereas if you have a very low value of K, you have quite good detection. So for instance, drop-seq type methods usually have a K above 50, whereas SmartSeq 2 methods usually have a K less than 30. That's a sort of a useful meaning there. Now we're going to move on to the next section, which is pseudotime analysis. So previously we looked at clustering, which assumes you have nice discrete states that are different from each other within your cell type. So you have one cell population that is all homogeneous within itself, but is different from another cell population which is also homogeneous within itself. Whereas pseudotime is what happens when that's not the case. So when you have a continuous transition from one cell population to another cell state, and you don't have a nice, clean dividing line between them, is when you start needing pseudotime analysis. So it's quite often used when you have some sort of differentiation process. So if you have a stem cell differentiating into any other kind of cell, usually use pseudotime analysis, as well as in sort of development, particularly in the embryo. However, the problem with all of these studies is that first, that the cells are not all perfectly synchronized with each other, and also that you can never sample the same cell twice. As soon as you're sampling a cell and doing an RNA-seq in it, you have to destroy the cell in the process, so you can't actually get a true time series. Instead, we get sort of snapshots of the population at different time points throughout the process, and the cells within each population is not perfectly synchronized with each other. So if you have a snapshot sort of in the middle of the transition from a stem cell to a neuron, some of those cells are going to look much more like the stem cell, whereas others will look much more like the neuron. So again, using just our sampling time points, we're not going to get a very good mapping of this transition. Instead, we want to infer the developmental time of the cell and then reorganize the cells in each of the time points along this new axis that's called pseudotime. So we're going to consider 3 different methods. The first one is T-scan, which actually combines clustering with pseudotime. So essentially, it takes all of your cells, does a preliminary clustering using mClust, and then builds a minimum spanning tree between the different clusters, centers. So you have a whole bunch of different cluster centers, and it draws lines between those cluster centers. And whichever branch of that tree has the largest number of clusters on it is defined to be the main branch and is used to calculate the pseudotime. So we're going to be using the STENG data set, which is early development of the mouse embryo. So starting from a single cell zygote, a very early 2-cell embryo, and then to 4 cells, to 16 cells, and then to the blastocyst. So in this data set, they sampled 10 different time points along this development. So we're going to use 10 clusters to begin with, and then we can run T-scan, and it produces this nice plot. So here we've got each of the different clusters is labeled, and then this main branch connecting all of the clusters together is defined to be the pseudotime. So in this case, we don't have any side branches coming off the main branch here. So then we can extract the ordering of the cells, and then we can look at which time points have been placed into which clusters. So here's cluster 10, which is mostly made up of mid and late 2 cells. So up here, that's the mid to late 2 cells. If you check some of the other clusters, hopefully that one will be early 2 cells. Hopefully that one's 4 cells, maybe 8 cells, and then somewhere over here we should get 16 cells and blastocysts. So we're going to plot the pseudotime ordering defined by t-scan. And then here we've got the known sampling time point, which I've also highlighted in colors, where red is early, so that's early 2 cells, and then purple is the late blastocyst. You can see that most of the known sampling time points have been defined as different time points by t-scan, whereas a couple of the later time points are sort of mixed together. Another method that's based on a similar idea is Monocle. Only Monocle skips all of this clustering nonsense and just builds a minimum spanning tree connecting all of the cells from the get-go, and then identifies the longest path in that minimum spanning tree, which it uses as the pseudotime, and branches coming off of that tree can be diverging trajectories from different cell types. So if you have one main branch and then one side branch, you've got one cell type that differentiates into 2 different cell types. However, Monocle does not identify the number of different branches itself. You have to specify that using the numpaths argument. The main disadvantage of Monocle compared to t-scan is that Monocle is very slow and will not run if you have too many genes in your matrix. So you first have to identify the important genes, which we did in the last section using M3 drop. So here you can see we get a nice several thousand differentially expressed genes using M3 drop, which we then subset from our whole expression matrix, and then we can run Monocle. Where again we have to first construct an object, and then it'll do some filtering for us. Then it does some dimensionality reduction, similar to clustering methods, and then produces a nice ordering, and then we can plot the tree. So here's our nice tree. You can see we've done dimensionality reduction to put this into 2 dimensions. And you get this one branch of the main tree, and then we get some offshoot branches there, and over here, and over here. But because we've said we only expect one path, Monocle has not defined any branching points. If you go up here and change the number of paths to 2, you should start getting branching points being defined. So then we're going to extract the ordering and store it in a somewhat more convenient fashion, and then plot the pseudotime versus the known time points. So again, we get very nice ordering of the cells according to the known time points, except for this last stage of blastocysts in this case, whereas all of the other stages are quite well defined. The third method we're going to consider are diffusion maps, which is actually quite similar to t-SNE in that it's mostly a dimensionality reduction method, also similar to PCA, only instead of t-SNE just tries to preserve local distances, and PCA assumes that all your dimensions are linear. Diffusion maps don't make either of those assumptions and just tries to fit a wiggly line through your data as a main axis of your main component of variation. It can be a wiggly line through your data. And it uses eigenvalues and eigenvectors and diffusion process, which is all this mathematical framework that makes it very efficient to do so. So again, this is how you run the diffusion map, which has to be on log transformed data, using this destiny package. And here you can see the two components that the diffusion map has produced. You can see that the first diffusion component corresponds to the pseudotime, with the early cells over here and then the later time points over there. But you can see that the diffusion map very well is quite good at defining the early time points, whereas the late time points are all kind of smushed together. Which is not terribly very good. So there's a few exercises that I'll just finish talking about all this stuff, and then I'll give you some time to work through these exercises. So now we can compare T-scan and Monocle. Unfortunately, the diffusion map package is still in development, so it's quite hard to extract the pseudotime at the moment, but hopefully in a couple of months that will be finished, and we can compare that one as well. So here's the ordering for Monocle on the x-axis and the T-scan order on the y-axis, and again the colors are the known time points. You can see that for the early stages we get very good consistent results, whereas for the later stages there's a lot more differences between the methods. Both T-scan and Monocle also allow you to look at the expression of different genes through the pseudotime in very convenient ways. So this is the function that you would use in T-scan to do that. So here we've got a gene called OBOX2. You can see it's most highly expressed in sort of the early-ish stages, and then lowly expressed in the later stages. And we display the same gene using Monocle, and again we get this nice humped curve. Yep.
So I don't understand this. So if you know the time points or the different stages, why do you need to do this?
So this is a very clean data set where the time points are very distinct. So we wouldn't need to do pseudotime for this data set necessarily. What you usually see, though, is, so if we go up to here, is that the known time points wouldn't be this clean. You would get many genes sort of in this late time point that wouldn't have finished differentiating completely, and would actually be placed further upstream, whereas some of these early ones might have differentiated much faster and be located further downstream, which is what you might most often see with particularly differentiation from stem cells to something else, whereas development tends to be a lot cleaner, where we know the time points much better. Any other questions? Okay, I'll give you a few minutes to work through the exercises on this page before we move on to the next section.